Welcome everyone. Welcome to our 10th seminar of the Aphasia CRE seminar series. I'm Caroline Baker, co-facilitator of the seminar series. I would like to acknowledge the Indigenous elders past and emerging and pay respects to the traditional custodians of all the lands on which we meet today. We warmly welcome you to this very special seminar, the first to cross international borders between the USA and Australia, while gathering hundreds of attendees internationally. We also acknowledge the difficult time of COVID-19 for everyone around the world. We are thinking of healthcare staff in our hospitals and communities serving patients, clients and families. We are so delighted to have Professor Swathi Kiran as our presenter today. Professor Kiran is Professor and Doctoral Program Coordinator of Speech, Language and Hearing Sciences and Associate Dean for Research with Sargent College of Health and Rehabilitation Sciences at Boston University. Before I formally introduce Professor Kiran, let's cover some housekeeping. This is our first seminar across countries using Zoom, so please be patient with us and the technology today. Our apologies in advance if there are any disruptions to your viewing during the seminar. To enhance everyone's Zoom experience, your microphone has been muted and your video turned off. Please also minimise your Zoom gallery so you can view the slides as best as possible. Thank you. Next slide, please. This seminar is being recorded and will be available to access along with past webinar videos via the website. Just click on the Resources tab. Videos are usually uploaded 1 to 2 weeks after each seminar. We'd also like to add that the CRE has developed a repository of the aphasia-friendly COVID-19 resources. These have been sourced from across the world. You are welcome to use these resources in your practice. Questions for this seminar will be aided by the use of Slido. Feel free to log in now so you can become familiar with it. You can log in anonymously, or we encourage you to add your name and ask a question. Our event code is 2509. Enter your question under the Questions tab at any time throughout the presentation. You will also be able to see questions asked by other audience members. Someone may have already posted a similar question to yours, so please double check. Like a question by clicking on the thumbs up. Questions can be upvoted to show those of most interest to the group. It's a great way to engage with the presentation experience, and Professor Kiran will answer as many questions as time will allow at the end of the presentation. Please engage with us on social media via Twitter and Facebook. Feel free to tweet along today and use the hashtag AphasiaCRE. Please join us as a member of the Aphasia CRE. We welcome people with aphasia, their family, friends, health professionals, researchers and organisations to our community of practice. Benefits to members of the community of practice include a monthly newsletter, updates about events and activities, contributions to research, networking opportunities and more. The CRE is always looking for financial support. If you wish to donate, please see our website for details. It is now my absolute privilege, and I'm so delighted to introduce Professor Swathi Kiran. Swathi Kiran is James and Cecelia Ying Professor of Neurorehabilitation in the Department of Speech and Hearing Sciences and the Associate Dean for Research at College of Health and Rehabilitation Sciences at Boston University. Her research interests focus around lexical semantic treatment for individuals with aphasia, bilingual aphasia, and neuroimaging of brain plasticity following a stroke. She has over 100 publications and her work has appeared in high-impact journals across a variety of disciplines, including cognitive neuroscience, neuroimaging, rehabilitation, speech-language pathology, and bilingualism. She is a fellow of the American Speech, Language and Hearing Association and serves on various journal editorial boards and grant review panels, including at National Institutes of Health. Her work has been continually funded by the National Institutes of Health, National Institute of Deafness and Other Communication Disorders, and American Speech, Language, Hearing Foundation. She is the co-founder and scientific advisor for Constant Therapy, now owned by the Learning Corporation, a software platform for rehabilitation tools after brain injury. Welcome and thank you, Professor Kiran.
Thank you. Shall I get started?
Yes, please. Thank you.
Thank you very much, Caroline and Miranda, for the invitation. I really wish I was there in person. I was really looking forward to coming to Melbourne in Australia in about a week. But COVID has disrupted a lot of our plans. So it's actually great to be here on this cross-Atlantic Zoom conference. And I look forward to sharing some of the work we're doing with you right now. So I will first start, let me make sure. I will get started with, as long as you can see my slides, I will get started with the disclosure first and just tell you the funding that I've received and the scientific disclosure. Sorry, there's a lot of different sounds coming from my phone. I don't want to change anything at this point. So if you hear a couple of tugs, just ignore them. Okay, so I want to start this presentation with a bit of a context. So a couple of days ago, about a week and a half ago, on Twitter, my colleague Julius posted a tweet saying, I was complaining to my PhD mentor, Audrey, about the lack of progress improving aphasia treatment in the past few decades. And her reply was that the researchers that know people don't know brains, and the researchers that know brains don't know people. I think she's onto something. And I think that this is a very timely post and chat. And what is interesting is that there's a few of us, excuse me, let me just make sure that. Okay, so I wanted to just start this conversation with a very, very current context where I think it is up to us to think about the brains of people who've had a stroke, as well as the folks who need the kind of rehabilitation that they have to receive after they've had a stroke. So what I'm going to try to do today in this talk is to give a very broad view of the work that we are doing at Boston University. And I've sort of, this is the outline of my talk. I'll give you, this is a roadmap where initially I will talk about the work that we're doing to examine markers, neural markers, brain markers, language and cognitive markers, and effective treatments. Then I'll talk about how this relates to predictive modeling of behavior, and ultimately the goal here is to come to develop better treatment outcomes and prognosis. So the work at Boston University is sort of in terms of the kind of factors that predict outcome have been lumped into three buckets, if you will. We're first trying to examine the degree to which brain damage influences recovery. In the second bucket, we have spent several years trying to examine the nature of language and cognitive impairments that influence the inability to function successfully in an environment. And finally, we've also spent many years developing effective treatments for specific language and cognitive function. more recently, and that's really what I'd love to share with you today, is our work to integrate these multidimensional factors into machine learning and computational models to predict which of these factors are actually important in predicting recovery, and how these pieces of information can enable us to produce better treatment outcomes and prognosis. The goal is to really get to personalized and individualized medicine and rehabilitation, because we know that one size does not fit all in aphasia rehabilitation, and we've been trying to take a data-driven approach. So in the first part of this talk, I will focus on some of the recent work we've been doing, looking at brain imaging parameters, language markers, and I'll tell you a little bit about recent studies we've been doing in terms of effective treatment. And then I hope to spend the last 1/3 of the last half of the talk on really recent work we've been doing in terms of predictive modeling. And they come in very different varieties of work that we're doing. And then ultimately try to convince you, I hope, that this is one approach that can lead us to better treatment and outcomes for patients. Okay, so we'll first look at the work that we're doing in terms of neural markers. And this is a very broad term because we have been looking at neural markers in several different ways. The upshot, really, the take-home message here, if you don't hear this by the time I get to the end of this section, is that we're really thinking about the brain of a person who's had a stroke as involving networks of regions. And different patients have different types of network organization, and that drives them to respond to treatment differently. So to sort of start this journey, I want to put some context for what aphasia recovery looks like. What does neural recovery look like after a stroke? And so this is a figure that we put together essentially that draws from work from R.J. Hillis and her colleagues, as well as Dorothy Sauer and her colleagues. And what this figure is showing is that in the acute phase, which is days and hours and days right after a stroke, what is seen as a reinstatement of functioning in that ischemic penumbra, the temporarily compromised left hemisphere, as well as restoration or reperfusion of blood flow, and that's key to recovery. In the subacute phase, that's weeks or months after a stroke, what we see is reorganization of the brain structure and function relationships at the level of brain systems. And so that's what you see here in the gray box. And then in the chronic phase, which is down here in the months and years after a stroke, what is really important is the establishment of new pathways and compensatory mechanism for lost functions. And this may be in different parts of the brain. What's overlaid on this are findings from Dorothy Sauer and colleagues, and they just updated this figure, their findings actually in a paper that just came out a month ago. And what they found was that greatly reduced activation of the remaining left hemisphere regions is really followed by an upregulation of right hemisphere homologs in the subacute phase. But in the chronic phase, you essentially see a normalization of left and right hemisphere activation. So this is what we know about recovery in the acute to chronic phase, but we've really been trying to focus on what happens in this dark blue phase, which is the real chronic aphasia. And here, I just want to step back to a study that we did in 2016. This study included only 17 patients. And in this paper, Jordan Sims looked at specific regions of interest that were damaged in patients who had a left hemisphere stroke. And the first thing we found was that left inferior frontal gyrus, pars opercularis and triangularis, despite significant damage across these patients, were the regions that correlated with behavioral accuracy on an fMRI task involving semantic processing. The next thing that we observed was that the more damage there was to these regions, specifically in the left hemisphere, like the LIFG, LMTG, and the angular gyrus and supramarginal gyrus, the higher the percent signal change or bold activation in bilateral SFG, MFG, and anterior cingulate. So these regions here. So the more damaged these regions were, the more active a bilateral network of other regions were involved in function. And then when we looked at both signal activation, so just fMRI activation, while we found that healthy controls had a left lateralized network, which you would expect, the patients were recruiting a very bilateral frontal network, essentially suggesting that this is not just a left to right question. There's really a bilateral network. It's highly driven by these frontal regions. And so this was back in 2016. And from this point on, we started to look at more carefully what did damage to the left hemisphere regions do to function. So in a paper that came out last year by Erin Meyer, we looked at 30 patients with aphasia, and they actually ended up getting treatment, which I'll tell you much more about. At baseline, they underwent T1 and DTI scans, as well as a battery of language assessment that we typically do. They also completed semantic treatment and semantic-based treatment to improve their naming function. And what we calculate from that is the proportion of maximal gain, is how much gain do the patients show based on what their baseline function is. And so this was a very data-driven approach. So in the first analysis, what Erin did was try to look at two dimensions, lesion location and lesion volume, in terms of these data-driven clusters. And you can see the black circle here is actually patients with a fairly large lesion, and the blue circles are a different cluster. Well, and in a different metric, she also looked at white matter metrics. So this included both gray and the underlying white matter tracts. And found, again, 4 different clusters that essentially captured different types of patients. And then using these different clusters of patients that fell on these 2 different dimensions, she looked at how these different clusters relied on how they predicted WAB-AQ, the BNT score, as well as this proportion maximum gain. And essentially, a long story short, what she found was that there were significant differences in the patients in terms of where their lesion sizes, their gray matter damage, fell on a continuum, as well as their scores on the WAB-AQ, the BNT, and more importantly, on this proportional maximum gain. The results were somewhat similar for whether we were just looking at gray matter damage versus gray plus white matter damage. But essentially, when we looked at correlations between these metrics and gray plus white matter metrics, what we found was for overall aphasia severity, there was a significant correlation between aphasia quotient and all of the gray matter and white matter metrics. Once we didn't control for lesion volume, but once we controlled for it, these relationships went away. So overall lesion volume seemed to capture a lot of that information. But when we included lesion as a covariate, we did find existing relationships that survived multiple comparisons between BNT and the fraction anisotropy in left ILF and left IFOF. So these are relationships that you can see right here. And essentially what was concluded in this paper was that preserved left hemisphere regions predict language severity and treatment outcomes. And whether you look at just the gray matter metrics or the gray plus white matter metrics, predict how impaired a person is going to be at baseline, but really only the gray and white matter metrics, the degree of gray matter and white matter integrity, predicted treatment outcomes, which makes sense because the connectivity of these tissues matters as well. So this study only looked at structural metrics of gray and white matter. In the next paper, we tried to bring in the fMRI activation as well as a measure of connectivity into this analysis as well. So here we're trying to really build this hypothesis that it's not just how much damage you have in the brain, it's also how the undamaged or active brain regions talk to each other, especially when someone's doing an fMRI task. So in this particular paper, we had a very extensive hypothesis that we laid out where we thought if you have no damage to the left hemisphere, you will see very strong connections between in left hemisphere regions, but as the damage increased, either from the posterior region or in the anterior region, all the way to complete left hemisphere damage, there would be shifts occurring towards the right hemisphere. So that's what these arrows show. So what I want to just emphasize again, in case you missed it, is in this case, we're looking not just at damage as a lesion, lesioned tissue, but we're also looking at fMRI activation as a measure of what is functioning in real time. So this study also had 30 patients. They all had the same treatment, T1 fMRI. T1 scans as well as an fMRI task. And here the fMRI task is a semantic feature verification task. So they see these pictures and they have to decide semantic attributes. And what we do from there is we then extract activation and connectivity using dynamic causal modeling to essentially look at relationships, synchronous relationships or effective connectivity between regions in the brain. So again, as you can see in healthy controls, the brain regions that are talking to each other in order to perform this task happen to be mostly in the left hemisphere, left IFG tri, LMFG, and so forth. And in the patients, there's quite a bit of bilateral connectivity that's happening between these brain regions. So this is the fMRI piece. But really what we were looking at was trying to then build fMRI and lesion into one set of explanations. And so this figure now shows how, where the lesion is and how much activation there is in the brain in the context of that lesion can be explained across these patients. So this is essentially a set of principal component analyses that break the data down into relationships between the degree of lesion as well as the amount of connectivity between regions. Now, this is a busy figure, and I won't have time to explain all the different parts of the way this the data played out. But to summarize this bit, what we found was that, of course, as we expected, lesion volumes were associated with, in this particular case, reduced frontal connectivity, which is a bit surprising because there's something about this frontal temporal relationship depending on where the lesion is. We also found that the greater the lesion volume in the left hemisphere, there was a greater connectivity between the temporal regions we looked at. So the residual regions were even more connected. And greater damage to left frontal regions, as we expected, as you can see in this figure, there showed shifts to the right hemisphere. So there was greater connectivity in the right hemisphere in the patients that had greater damage in these frontal regions. So ultimately, our conclusion was not as clean as what we show in this particular figure, but for sure, we found connectivity shifts to the right hemisphere and inter and hemispheric connections depending on the degree of damage to the left hemisphere. So what this is really, again, pointing to is that it's not a left versus right issue in terms of recovering the brain. It's much more nuanced, and it also depends on what sort of shifts can happen towards the undamaged tissue in the left and right hemisphere. So from that work, we then are starting to look at slightly more connection-based approach. So this is work that we're just finishing up right now. Here, we're looking at the degree of disconnection, which is the opposite of the degree of connectivity. This is a structural study. In this particular study, we've looked at 76 patients. They all get a baseline MRI scan and a whole bunch of language assessments that I'll show you in a bit. And what we're calculating here is something called a disconnectome map. And essentially, and I can explain this in greater detail, essentially, we take the lesion map as well as white matter tracks from healthy controls to calculate what is a probability of white matter disconnection at every voxel in the brain. And then we look at relationships between these voxel-wise disconnections and language scores. So it's a little bit like voxel-based lesion symptom mapping, but it's not exactly that. And I can get into the details when we get to the questions. And so what we found here, before I show you the figures, is that as we expect, again, when we look at language impairment, aphasia quotient, and measures of language function, are significantly associated with disconnection in the left perisylvian region. So it's again, the flip of saying the integrity of the white matter predicts function. Here, the degree of disconnection really matters as well. And the core disconnectome maps are in left arcuate fasciculus and IFOF. And essentially in this particular analysis, we looked at aphasia severity, syntactic comprehension, semantic processing, syntactic production, as well as repetition and a couple of other measures. And what you can see in these figures are maps of the degree of disconnection that are overlaid across patients. So we take out parts of the map where everybody has a lesion, and now we're really looking at the degree of disconnection. And there's a lot of overlap across these patients. So of course, arcuate fasciculus and IFOF are quite damaged, but when we look at the ones that are disconnected, there's quite a bit of overlap across these tracts, including the uncinate corticospinal tract and left ILF. And now we're using these sort of these pieces of information to build into the predictive models that I'll tell you about later on. So that those are all studies where we're trying to see which sorts of measures are important for predicting either language impairment or some measure of language recovery. But we've also done treatment. We've done fMRI studies in patients before and after they receive treatment to see what exact kinds of changes we see in these patients. So in the first study, this was done by Jeff Johnson. We looked at 26 patients who all had naming deficits. They underwent the structural and fMRI task. Here we're looking at a naming task where they look at a picture and either say skip or they have to name the picture. And then they go through 12 weeks of a naming therapy, which is essentially a semantic-based feature treatment where they're asked to name pictures and go through the semantic attributes of that. And what we did in this paper was we looked at brain activation before and after treatment, and we divided this into specific ROIs of interest in the left hemisphere, sort of going from the paper I described earlier by Jordan Sims. And while we found a increase in activation as a function of time, post versus pre, what we really found, which was not surprising but really disheartening, was it was really all over the place in terms of results. But actually, what is interesting in these results, I'm pointing you to this graph at the bottom, and what you can see on the x-axis here are different regions of interest: LIFG, ORB, TRI, OP, LMFG, PCG, NTG, LSFG, angular gyrus, and then the right hemisphere counterparts of these regions here. And healthy controls mostly show deactivation in these regions. This is average percent signal change, so it's negative for the healthy controls. And what we see for the patients is that there's a decrease in some of these regions, especially in regions such as the LMFG and so forth. But then there's an increase after treatment in many of the right hemisphere homologs. Well, that makes perfect sense. So we could just pack up and say, well, it's all about the right hemisphere activation. There's, as a functional treatment, you see more activation in the right hemisphere, we're done. But we didn't actually stop there because we knew that this was a little surprising to see a decrease in activation in some of these left hemisphere regions. So we dug in a bit deeper and we looked at the degree of damage in these regions, because we could calculate that. So we were able to calculate percent spare tissue in any given region and how much activation we were seeing in the remaining tissue. And we were able to look at how much the percent signal change changed before and after treatment. So here, the left-hand side are pre-treatment percent signal change, and on the right-hand side are post-treatment percent signal change across these left hemisphere regions that we know are very damaged to varying degrees in these patients. And the way this graph is organized is the ones at the top actually show an increase in activation, and then the ones at the bottom show a sort of decrease in activation as a function of treatment. And this is really the main story, is that the degree of change that you see in undamaged regions, so these are the remaining residual tissue, depends on how much damage there is. So while we can conclude from these graphs is that there's an upregulation of traditional language regions, like especially in the right hemisphere homologs, and a couple of these regions like LIFG orb. What's really important is that the degree of activation in these paired tissues is really dependent on the amount of residual tissue there is. So it's not a straight linear relationship. And so this is very important to us right now, because we're starting to build network models, and it allows us to see how much the degree of spare tissue in a brain region matters. So from this, while we concluded that there were positive changes in activation as a function of treatment, we wanted to dig further into, what exactly are these individual differences? Why do some regions engage more as a function of treatment, even though they're damaged, versus others don't? So in another paper also by Jeff Johnson, we started to look at the brain in terms of network regions. And so I'm actually making a bit of a bigger jump here. And I can come back to that jump I just made, but now we're actually going to talk about patients who've received these treatments, and we know that they respond to treatment differently. And we're starting to look at their brains in terms of network features. So it is these 26 patients who I just talked about. It's like different, maybe a couple of them are different based on the data we had, and they all underwent these T1 fMRI scans and went through 12 weeks of naming treatment. Here they're performing a semantic feature judgment task, which is not the same as naming, but it's a different semantic task. And they all got the semantic naming treatment. Now, what we did in this study, before I show you the results, is that we divided these patients into responders and non-responders based on how well they did in the semantic feature analysis treatment. And I will show you some more results later on. But we ended up with two groups of patients in this, so it's roughly 17 and 11 or something to that effect. And then we looked at how the brains of these patients were different before treatment. So pre-treatment graph theory metrics. So we looked at brain regions such as different regions that I showed you in the previous slide, but in terms of the global and nodal local efficiency. And what we found here was very interesting. What we found was that when we directly compared the patients who responded to treatment versus the ones who did not respond to treatment, we found significant differences in specific nodes in the brain in terms of the node strength, of the degree of and the degree of the nodal connection, the degree of connectivity these nodes have, in terms of global efficiency as well as local efficiency. So let me just orient you a bit to these graphs. What you're seeing here are different regions, and each of these calculates a node and an edge in terms of how they talk to, what sort of connectivity they have with other regions. And the ones that are in red are significant differences between the responder group, so higher in the responder group than the non-responder group. And right away, you can see that in terms of node strength, you've got these few hubs in the right hemisphere that are higher for the patients who ended up being responders than the ones who did not respond to treatment. There are some left hemisphere regions also involved in global efficiency, so that's as well as local efficiency. And all of these essentially point to the fact that patients who had higher network strength and global efficiency were the ones that showed more changes, and somehow something about their brains were ready to take that treatment than patients who had lower strength and lower global efficiency. And what this is leading us towards, at least this is the first time we've seen this, and we're starting to look at this even more, is that the relative preservation of global and local efficiencies, how well these brain regions, these undamaged brain regions talk to each other, it may be a biomarker for treatment-related recovery, because these are conditions of what the brain looks like even before they receive the intervention. So all this work is ongoing. I just gave you snippets of studies that we're just finishing up We're finished in the last couple of years, but we're really trying to lead all this towards a theory of what we think might be a framework for recovery, especially as a function of rehabilitation. So here we are building from a model that was developed by Heiss and Peel back in 2006, where they suggested that there's a hierarchy of recovery with increased damage to the left hemisphere. First of all, and I think this is a no-brainer, that we really think that there's a network of regions that are activated in the service of language recovery. I showed you that in the Johnson paper where we looked at the ROIs. What we think is that to the extent that left hemisphere regions are spared, they're extremely important and engaged in language recovery. Again, but all the slides I showed you, the last couple of slides I showed you essentially show that. But as damage increases in the left hemisphere, activation shifts to the right hemisphere, and as well as multiple demand and domain general regions like the MFG. And then what I didn't point out to you in the Johnson cortex paper, what I showed you, the differing percent signals changes, is we actually see activation in regions like MFG, which is not particularly a language region. So there's something about these domain general regions that are kicking in for language recovery as well. And finally, this is just work we're starting to do, but the degree to which this network typology instantiates determines the extent of recovery. So patients with efficient typology show greater improvements than the patients with inefficient, which is the slide I just showed you before. So we don't know if this is all true yet, but we're starting to think that when somebody has a stroke, right after the stroke, of course, there's a lot of damage. There's quite a few studies that show that there's dysfunction in the network characteristics of these brains. But as they continue to recover slowly over the weeks and months, patients who have better reorganization or better network topology show more improvements. Their brains seem to be ready for the kind of therapy we give them, and as they respond to that therapy, their brains look more and more like healthy controls. But then there are patients who don't have this efficient functional connectivity and efficient network topology, and their brains don't actually they don't seem to be responding to the kind of treatment we're giving them. This is all work we're just starting to do now, so we don't know how much of this is true, but at least it gives us something to test. In addition to all this, we also are trying to think about principles of neuroplasticity and in ways that would matter for intervention. So those are not surprising things, but the fact that we need to use it, improve it or lose it, and therapy builds targeted networks. Repetition is important, and we promote generalization and avoid interference. And lastly, about complexity. So there are other principles in here that matter in terms of the kind of plasticity we'll see, but we're starting to look at all this. So that was just the work that we're doing in terms of neural markers. We've spent a lot of time the last few years looking at this, so I will have to reduce my time for these other two aspects because just because I see the time, it's already 7:40. So in terms of language markers, again, I think we've done, I would essentially give this very high level framework of how we are very interested in the link between language and cognitive functions, such as attention, executive function, cognitive control, and learning. And I will not have any time or do justice to the work that we've done in all of these regions, these topics, but I point you to some papers from some really outstanding students in my lab published in this area. I do want to spend a little bit of time on this idea that we think language, attention, executive function are all sort of part of a bigger umbrella of cognitive language and cognitive function. And this is really work that we have done trying to predict, trying to see what aspects of language function and what aspects of cognitive function predict treatment outcomes. So this is a paper that came out in 2019. where when we first looked at the kinds of behavioral measures you would calculate on the WOB and the PAPT and the WAIS and so forth, we essentially came up with two composites, a language composite and a cognitive composite, that predicted our proportional maximal gain, which is our measure of treatment outcome. That's not surprising because all that says is language function predicts treatment outcomes, cognitive function predicts treatment outcomes. But in a follow-up analysis, we said what exactly in these cognitive functions predicts this proportional maximal gain. And what we found was that executive function and visual short-term memory predicted those treatment gains. And then when we looked at the maintenance of this function, these same measures significantly, again, executive function and visual short-term memory predicted treatment gains. So this is an indication that we do need to think about a patient's cognitive function in addition to their language function in terms of how well they would predict whether somebody would benefit from treatment or not. So that's just one study I'm going to talk about since I don't have time to get into too much detail there. I do want to spend some time telling you about the kinds of treatment studies we're doing right now. Again, we have in the past looked at many different types of impairment, psycholinguistic approaches, and also gotten into functional changes in these measures. So I won't have time to mention any of those. But I want to talk about 3 recent treatment studies we have finished in the lab to give you a breadth of how we're doing this. So in the first study, this is a naming therapy study. A lot of the therapy I talked about before. There are 25 patients in this paper. They're getting naming therapy for 12 weeks. And they're either getting therapy for typical examples in the category, or they're getting therapy for the atypical examples in the category. And I showed you the slide before, but the therapy is essentially a semantic feature treatment where they're working on features of a typical example or an atypical example. And what we found in this study was that when we compared the degree of acquisition between the trained items and the untrained items, all the patients improved much more on whether they were trained on the typical examples or the atypical examples compared to the monitored. So what they're trained on, they improve much more than something that you're just tracking over time. But what was really important about this study, and I think really something that we have to all come to terms with, is not all patients responded the exact same way. So when we divided the patients into the degree of responsiveness to treatment, all of those effects that I just showed you in this previous graph were driven by the patients who responded to treatment. The non-responders are essentially showing absolutely no effect. And this is important because what it's telling us is that the therapy does not work for everybody. And these were the criteria that Jeff used in his study to divide between the responders and non-responders. The other thing we were looking for was generalization, whether training atypical examples matters or training typical examples matters. And again, here what we found was that the degree of generalization to the typical untrained examples was higher than for the atypical examples. So the atypical examples generalized more to the typical than the other way around. But again, the results are only true for the responders, and the non-responders don't show any of these trends at all. So that's one study that we've done. The bigger take home from the study, when we looked at the rest of the test scores, were that responders show very different acquisition and generalization patterns than non-responders. And this is something that we're looking at in the brain as well. In another study we just finished that's under review, we looked at the VNS treatment in Mandarin English bilingual patients. As you know, we're doing a lot of work in bilingual aphasia. So here, Jean Lee took Lisa Edmonds' VNS treatment that was actually done in Korean by a different group, and converted that into Mandarin Chinese, following the exact same steps, where they would do the verb and the thematic role assignments in Chinese. And then we had a sentence task, which was a probe, and a control task. What we found was that it's just 2 patients, but it's a preliminary, and it's a very preliminary study, but we only trained Chinese in these patients, and we looked for generalization to English. In the first patient, there was only acquisition, very little generalization, but I think the effect sizes were actually significant. when she did the analysis. And the second patient, there was quite a bit of improvement on the trained set and the untrained, but there was improvement across the board in English as well. So this is the first study that's looking at cross-language generalization from English to, from Mandarin to English using this VNest. And then the last study that we're currently doing right now, which I'll tell you a bit more about when we get to the predictive model part, is treating Spanish-English patients on a naming treatment. So far, we've only collected data from 13 patients, and these patients all undergo 10 weeks of naming treatment. They either get the therapy in English or they get it in Spanish. And again, here, what we're looking at is improvements on the trained items, as well as cross-language generalization. So a patient could get trained on the word sheep, and we're looking for generalization from sheep to oveja, or the patient could get trained on cabra, and we're looking for generalization from cabra to goat, or cabra to oveja. The treatment here also is a semantic feature-based treatment, with the treatment slightly different here. We have them actually produce, in addition to doing the 6 features, we have them produce it in a sentence target as well. And so far, at least, the results, the study is by no means done, but just based on the data, it looks like we're getting good treatment effects and some generalization effects as well, but not as strong. This is an ongoing study. We're hoping to enroll 48 patients. Because of COVID, we're doing this all remotely online, so it's quite the challenge right now. And we're trying to see what factors predict treatment outcomes. So these are just 3 of the studies that we're doing right now. And I think I'm going to skip these 2 slides because I want to have time to talk about, oh, I have to do that. Sorry, that would take me a while to explain. But these are just a few of the studies that we're doing to see whether treatments work or not. And so far we know that the treatments work, but we really don't know which treatments work for which specific patients. And then the goal is to standardize and personalize that intervention. As I said before, COVID has forced us to get our therapy online and remote, but even before COVID forced us to do that, we were already trying to deliver a lot of the therapy online, because we know it's virtually impossible for patients to come into the clinic or the hospital in places like Boston. So that's a big part of what we've been trying to do. This allows us to not only go back to the predictive modeling piece, which I'll tell you in a second, but also get to that individual patient. So the goal is that by delivering therapy remotely to a lot of patients, we can start to do population level analyses, but we can also start to look at how small cohorts of patients respond, as well as getting down to that individual patient. So this has really been an amazing opportunity to do this predictive modeling with really large sets of data. And to do this, we've been using the constant therapy software platform, which that I co-developed many years ago. And I'll skip this slide because I think you probably know what constant therapy looks like. There are various therapy tasks that patients practice on their iPad. But the more, and there's a bunch of studies that you can go back and look at that talk about the efficacy. The real piece to this is that because it's deliverable on an iPad, and pretty much anybody, including people in Australia, anybody who speaks English can download this app and use it. We've been able to collect thousands and thousands and thousands and thousands of data points. And that's allowed us to do a lot of very careful predictive modeling of the data. And again, I won't have time to go through all the details of the kinds of predictive modeling we're doing, but we are able to essentially sift these millions of data points into factors such as age, lesion location, months post-stroke, and more importantly, factors such as the amount or intensity of therapy, how many times do patients practice, what's the optimal dosage, what type of treatment are they doing, do we see generalization from language to cognitive function, and get at again, what might be these personalized therapy outcomes for patients. So that's one big piece of work. I'm happy to come back to that if people have questions, but I do want to also get back to some of the other work that we just started to do. And this is the different types of predictive modeling. We don't have the luxury of those millions of data points we have for the constant therapy data set. But we do have imaging data on these folks, so it allows us to do a completely different predictive modeling. So this study I'm going to tell you about comes from 80 patients that we collected as part of the CNLR data from Boston University, Northwestern, and Johns Hopkins. And these 65 of them are who I'm going to tell you about, participated in naming sentence production or writing therapy, and we're getting a whole bunch of structural markers on them and a whole bunch of behavioral measures. And for this particular analysis, the dependent measure is who's going to respond to treatment. So here's just a schema of that. So we have a whole bunch of neuroimaging markers. We've started this analysis to just look at structural markers, and we're building the functional data into subsequent analysis. We have scores on WOB and a whole bunch of behavioral measures, and we're implementing this in machine learning models to then decide, can it predict who is a responder, sorry for the typo, and who is a non-responder. And to do that, we essentially then take all of these factors and put them into sort of sub-cohorts. So we have a demographic factor like age, education. We have a structural factor like how much spare tissue there is in different parts of the brain. And then we have behavioral factors such as WOB, AQ, and so forth. And then we build these random forest analyses that allow us to compare our data into specific sets of metrics. So is just demographic factor information enough to predict outcomes? Do you need to have demographic information and structural markers? Do you need to know everything about the demographic information and the structural and the behavioral factor? It turns out in this analysis that is quite true. And so just preliminary results that we got out this year show that the best model that predicts behavior is a combination of structural integrity in these deep domain general language regions and some language severity and the age of the person. And that's enough to classify whether somebody is going to be a responder or non-responder. But as I said, this is data that we're incrementally adding more and more features or factors into, and so we'll be publishing a lot of this in the coming year. So that's one way to think about predictive modeling. The other approach to think about predictive modeling comes from our work in bilingual aphasia. And this is really exciting because we have essentially developed a computational model that behaves like a bilingual person. So this is a semantic map, a computer semantic map, and an English map and a Spanish map. And this computer model essentially starts off like a newborn baby and then gets progressively exposed to more and more items in English or Spanish. And we can vary the age of acquisition as we want to the point that these maps look exactly like how bilinguals language book representation looks like. And once we have proven, and which we have, that this model can behave like any bilingual you could think of, we then introduce a lesion into the model, and we can either introduce a lesion at the semantic level, or we can introduce a lesion into the phonetic level. So we can either produce a lesion here or in any of these maps, and we can simulate what it's like to be a patient with bilingual aphasia. So they can have equal amounts of damage or relative amounts of damage. And we've done that also. And then we have then tried to simulate what it'll be like for this virtual lesioned computer model to receive rehabilitation. So the beauty of this work is that because it's computational simulation, we can predict for any patient what it would be like if you train the person in one language versus the other. Whereas in human beings, you actually never have that information. You only know which language you train them on, and then you get the results for that particular outcome. And then you think, oh, that went well or that didn't go well. In terms of computational modeling, we can a priori predict or simulate what the outcome would be. And more importantly, this tells us which language to treat the person in. So this is a different kind of prediction. This uses evolutionary algorithms. These are essentially neural networks, neural network type of modeling, very different from the random forest model I told you about before. But this takes into account all these different features. And what's exciting about what we're doing right now, which relates to the treatment slide, is that we're doing all this in the context of a double-blind randomized control trial. And so here our goal is to get 48 patients. They can either speak English or Spanish. After we finish the assessments, we give that information to the computer science colleagues who then compute these sort of simulations. And then based on the simulation outcome, they can tell us which is a good language to train. So here In this graph, you can clearly see which is the better language to train the patient. English, check. And they give us that recommendation, and then we go and train the patient based on that. So if the patient happens to be in the model prescribed group, they will get exactly the language and therapy that the model told us to do. And if they happen to be in the opposite group or the placebo group, they will get the exact opposite. So the model may tell us to train them in English, but we will train them in Spanish. And they go through all the treatment steps that I described a couple of slides ago. And then at the end of the study, we'll actually be able to say whether this sort of, computational modeling can predict the treatment outcomes better than the trial and error approach that we're doing. So again, this is work that's ongoing. I hope to share these results with you as well in the next couple of years. So these are just different ways that we've been looking at how to predict treatment outcomes, how to use different approaches of modeling. But as you can see, in all these cases, we're really trying to get at what's the best outcome for that patient, knowing fully well no two patients are the same. And the important thing here is to really take information about each patient's background and use that to predict personalized outcomes. The goal that we think we want to see in the future is that once any patient has a stroke and they go to a lab or a clinic, at the end of when they leave the lab or clinic, they will get a neural profile or a social language profile, social function profile, or a language cognitive profile workup. So it'll be part of the diagnostic assessment. And then all that information will go into one of these algorithms that we're trying to develop that can then predict that particular person's recovery profile. So they will say, based on the information we have here, based on the way we know these models can predict outcome, this particular person, number one, should get intensive individual therapy. Patient number two, based on the information we have, should participate in some individual therapy, but also in some social groups. And this person, the third person, actually should be involved in vocational training. So this is work that's just beginning, but I feel pretty optimistic that in the future, when a survivor of brain injury comes to a lab after they've had their initial recovery, we will be able to come up with these profiles and then use this information to predict that particular person's recovery. And by then we would have really learned to understand all these reams of data we're collecting and give them the personal recovery prescription that we think will be most useful. Okay, so I'm going to pause there and take questions from people. Should I stop sharing here because you take it back, Caroline?
Yes, thank you so much, Swathi. Kelly will share the screen. Thank you for fascinating and highly informative talk. We've just had so much engagement on questions, so we'll get through as many as we can. We will, if it's okay with you, Swathi, run over time a little to answer some of these questions. Is that okay?
Absolutely, absolutely.
Oh, great. All right. So Sam Harvey, one of our PhD students at the CRE here, has asked, is right lateralization of cortical activation in response to lesion a neurofunctional correlate of learned non-use? Is it good or bad for recovery?
Yeah, that's a really great question. I think what our data is telling us now, and hopefully some of the other labs, that right hemisphere activation is not bad for recovery. It seems to be important for recovery, especially during the subacute phase. And maybe even in the chronic phase, but it's not just anywhere in the brain. There seem to be certain regions, especially the homologous regions like right IFG, that are actually quite good for recovery. So there's another paper that I didn't have time to talk about today, but Dorothy Sowers, student Anita Stockard has similar findings that just came out in Brain that also gets at this question, that it's not bad for recovery, but it's not taking over recovery either.
Okay, lovely, thank you. Outside of the research setting, is it possible to get some information from MRI, not fMRI, the functional ones, regarding extent of damage and connections to hypothesize recovery?
That's a really great question, and that's really, the final slides I was showing is really where we want to be. Part of the initial slides I was showing you was our effort to try to get the minimal amount of information from a structural MRI, which I think most patients will have access to. That then you need to know, once you have this, when you can look at an MRI and you get some basic information, you have enough, with confidence, you have enough information to predict whether the patient's on a good prognostic track or not so good, and then decide what kind of intervention to give them. We're not there yet. As you can see, I've shown you slides that have all sorts of measures, and we don't know which one's most important yet, but that's where we're trying to go.
Okay, thank you. Are there any factors, for example, gender, age, psychosocial, or patterns you can report associated with strength of nodes in undamaged neural tissue?
That's a really great question. So in all the analyses that I showed in the first half of the talk, we use age and time post-stroke as covariates. They just don't survive any of the models because they get kicked out pretty early on. We don't use gender as much because it's a binary factor and we don't see as many differences. And in fact, there's another paper done by Georgina Angelopoulos where we looked at DTI tracks between women and men and how it related to language function. We found absolutely no differences between men and women. This paper came out last year. So we're pretty comfortable that these differences are not due to gender. They don't seem to be due to age and time post-stroke also, but they do seem to be about lesion severity. That's the thing that really props up to the top. In terms of psychosocial function, I have to say we haven't done a good job measuring that or using that as a covariate, but we're trying to change that now. We're trying to get better measures of participation, depression, motivation, and I hope those factors will be important. I would think they would be.
Lovely, great, thank you.
Yeah.
Sonia Brownset from the University of Queensland, she's part of our CRE. She asks, were healthy controls completing the same functional MRI task as people with aphasia? Could the differences in activations be due to the additional effort required by the people with aphasia?
Yeah, that's an excellent question too. Yes, in all the studies I presented earlier, we have healthy controls doing the exact same tasks. Which I just, in some cases, I showed you the healthy control data, in others I didn't. And I think it does, I think you're absolutely right. I think that a lot of the bilateral activation that we see, especially in the Erin Meyer connectivity paper and so forth, are due to extra effort by the patients. It's hard for them. Language processing is hard for them, and they engage more regions, and they show more activation in anterior cingulate and the superior frontal region, the medial frontal region. So that's absolutely the case. Now, to what extent that extra effort is actually helping them successfully complete the language task versus they're just really trying and they're not able to do it is what we're trying to tease apart now. So our current project right now, it's a collaboration with Fedorenko, is to look at domain general regions versus language regions to see to what extent these domain general regions are just sort of like waving and, it's like you're thrashing and trying really hard, and versus, and they would be involved no matter what you had the patient do, whether it was language or math or anything, versus how much of these are actually involved in language function and they're associated with better recovery. That's what we're trying to answer. We don't know the answer to that just yet.
Okay. Now, another PhD student here at the CRE, Maya, has asked, what do we know about responsiveness to therapy in people with chronic aphasia?
A lot of the data I was showing you where we were comparing responders to non-responders is actually all the data I showed you today is chronic aphasia. And when I mean chronic aphasia, I don't mean 9 months post-stroke. I'm talking about 10 years post-stroke, 15 years post-stroke. And what we're seeing is that some patients, as you saw some of the graphs I showed you, especially our treatment data, some patients are very responsive, even 10, 15 years after stroke, and they really do improve on not just what you're training them on language measures and functional communication, and others are not. And so that's exactly what we're trying to find out here is why are those people not responding to treatment? Is it just their brains, or is it more than that? I hope that by the time we make more progress on this, we're able to say that it's not just the degree of brain damage, it's also the motivation that they've lost over the years. And I hope that's the answer in the future.
Lovely, thank you. We might just take one more. Is it felt that the right hemisphere is taking on linguistic function, or that this increase reflects cognitive function reorganisation to support language?
Yeah, this is similar to Sonia's question. I think it is. I think all this data, especially the data that we're showing, where patients are showing increased activation as a function of treatment, where the activation is correlated with the degree of improvement they make in treatment, suggests that these regions are actually engaged in that improved language function because they improved in the treatment. Does it reflect increased reorganization? I don't know yet. I think that's what these network measures are going to tell us. It's definitely not, oh, the left hemisphere IFG is damaged, the right hemisphere took over and we're done. I think it's way more complicated than that. It's a dance that all these regions are doing with each other, depending on how much damage there is. And that may be cognitive function reorganization, but I don't know if that's exactly what I would call it. More work needs to be done there, but I think that the fact that these patients improve means that some of this is really change that's happening as a function of treatment.
Okay, great. John Pierce, he's a PhD student as well with our CRE, and
I don't mind taking all the questions if we only have 5 or 10 left. That's totally fine.
Oh, okay. Thank you. Yeah. That's lovely. Determining the relative contribution and interactions of all of these predictors must require very large studies, perhaps thousands. Is this the eventual plan?
I hope it is. And for that, so there's two things. The easy way that we're able to do this, as I pointed out, is in the constant therapy data set, where we've been collecting data since 2012. So we have at this point millions of data points. So that doesn't really take large studies. It just takes a large amount of data mining to figure out what factors protect. The imaging studies are totally on the opposite spectrum there. It takes a lot of time to collect the imaging parameters. And that's why databases like Plurus are so important, because they've been collecting data over time. But I also think that no one lab or one group can get this done. I think we really need to collaborate on these data collection measures, so that we are collectively collecting thousands of data points across our different units, and then we can get at these answers. But yeah, I think these do take a lot of data. Now, some of the machine learning approaches I talked about, such as a random forest approach, are a little forgiving in terms of the amount of data you have. They're not as stringent as the linear statistical regression models you would not run, but even they're not perfect.
Lovely, thank you, Swathi. Thank you also for being so generous with your time with answering these questions. Are you happy to keep going?
Yeah, we have 9 more, right? Okay. We can go through some of these.
Is there any correlation between handedness and interhemispheric reorganization?
Not that we've found so far. And that's what, so we always use handedness as another factor, and we don't see anything.
Okay. And Professor Linda Worrell asks, recovery or response to treatment is measured using the Boston Naming Test and the WAB. Do you have any plans to implement the core outcome set for aphasia?
Yeah, absolutely. So I'm very happy that we can use the WAB finally as one of our core outcome set. I'm probably one of the few people in the US that's pushing for these measures. And that's why I'm very eager to get some of these functional outcome measures into our core outcome set, so that we can start to look at predictions on those outcomes. So far, we've limited ourselves to treatment outcomes that we're measuring percent naming and so forth. But absolutely, that's our goal, is to run down the list of the core outcomes set.
Lovely, thank you. For those individuals who have low global efficiency and become non-responders, presumably there is increased risk of atrophy and vascular dementia in the future?
Oh my gosh, that's a really interesting question. We don't know, but if you're anonymous, but if you have data that makes you suggest something, please send it my way, because I think that that's exactly what we would like to look at. So there's a lot of correlations between poor brain health, poor outcomes, eventual dementia, but and these sort of low global efficiencies and so forth in stroke patients as well, but we haven't quite put all those pieces together. And I think you might be onto something here, and that's sort of what we're trying to study as well.
Right.
Great question.
Thank you. What are the characteristics of non-responders in the Gilmore study? Are these robust? Are they predictors?
Yeah, so I didn't have time to go over the details, but the non-responders in the study not only did not improve on the train measure, which was percent naming, but they did not improve on any of the outcome measures. On the WOB, we had a whole bunch of semantic processing tasks. They didn't improve on that. We had a whole bunch of CALPA measures that didn't improve on that. So they just, they really don't show any changes as a function of treatment. Now, we've actually, I didn't quite understand, are they robust predictors? I didn't understand that part of the question, but what we're trying to do is to see what predicts someone to be a non-responder. So we're sort of flipping that question the other way around and saying, what makes somebody a non-responder? Is it, as the previous person asked, their vascular status, or is it just a large lesion volume and that's it? It looks like when we look at individually those factors that all seem to be true, but we haven't put everything together yet to see what exactly matters there. But the fact of the matter is they really don't change on any measure, which is really depressing.
Okay. Sam Harvey asks about the Gilmore article in Archives, the cognitive linguistic test pre-treatment predictor of recovery. Cognitive function mediated by fatigue, motivation, etc. Could cognitive probes during treatment inform stronger models?
Yeah, that's a great suggestion. We haven't been doing that, but that would be actually work. That would be good future work to consider, is to also do those sort of probes. Because offline data tells us clearly their attention and short-term memory influences the outcomes. That's a great suggestion.
Yeah. Lovely. Sonia Brownstead asks, I really love that DG regions predict outcome in Gu et al. Should SLTs be considering these regions domains in order to augment therapy gain?
I mean, I would love to hear what your group's questions are, thoughts are about here, about this particular question. I feel like the data is heading there, but it's not there yet. For sure, domain general regions show up not only in our work. Cindy Thompson's group looked at changes in their sentence production treatment and found domain general dorsal attention network changing as well. So to what extent we need to be targeting these or making them a focus, hypothetically, I think absolutely we need to be thinking about that, but maybe we need some more data to see what exactly is changing in these domain general regions. A lot of people are looking at that, so I'm sure we'll find the answer fairly soon in the next few years.
In neuroplasticity literature, I have read about learned non-use and axonal sprouting. Do these relate to the connection and disconnection models you discussed?
Ooh. Yeah, that's a tough question. Yes, for sure, not learned non-use and axonal sprouting are neurophysiological mechanisms that underlie function changes. But I think a lot of those studies are looking at very, very short time windows right after the stroke or right after some extreme physical activity. So the time frame there is in the time frame, few days or weeks after the stroke model. What I'm talking about are, as I said, people who are 11 years post-stroke, 12 years post-stroke. I don't think the timeline lines up that well, but again, I don't know if years of non-use ends up leading to the kinds of disconnection we see. I don't know if that's true, but definitely I can't jump that bridge just yet because the timelines are very different.
Okay, thank you. Could there be other demographic, I think we've had a similar one, that influence response to treatment, such as low mood?
Yes, I think yes, and I'll take all suggestions for what people might think are different factors.
Okay. Sorry.
No, go ahead, nurse.
Do you think as SLTs we need to be pushing more in clinical settings for MRIs, functional MRIs, to be done with people with aphasia to inform our clinical decisions?
I don't know if we yet need to do that. I think we need to wait for the data to come out from a lot of studies that are going on right now, from the Plurus database, from Julius Fredrikson's work in South Carolina, from our work, and Miranda is starting to do this, David Copeland's doing this. I think we need to wait to see what all this data shows in terms of how important are these measures. And if they turn out to be important, yes. But so far, I'm not sure yet. I mean, how predictive they are. There are a lot of factors that matter, and this may just be one of them. But I think we'll have answers to that soon. That's why I said in the future, these things are important. Hopefully will be important.
Yes. Thank you, Swathi. We've come to the end of our questions. You've been so generous with your time and sharing your vast knowledge. We're just so appreciative. And just by the level of engagement we've had, it's been just a fabulous talk. So thank you so much. We really appreciate this.
Thank you, everybody.
Yeah. We've still got some people online, so I just encourage people to go to the polls tab if there is anything in the future that you would like to hear about through our seminar series. And we also look forward to hosting the next one on Wednesday, the 24th of June at 3 o'clock Australian Eastern Standard Time. And we are excited to have Dr. Richard Linley, Professor of Geriatric Medicine, present from the University of Sydney. He will discuss new thoughts about accumulated deficits, including aphasia in dementia. Thanks again, everyone, and we look forward to seeing you next month. And hopefully we get to meet in person one day, Swathi.
Yes, I hope so too, on this side of the world or that side.
Yes. Thank you so much.
Thank you, everyone. All right, bye-bye.
Bye.
Bye.